Hello there, and in this video we're going to be talking about osmosis and cells for iGCSE biology. We're going to start off thinking about how solutes dissolve. They dissolve in water by water molecules surrounding ions in solution. So let's take this Cl- ion here, or this Na+ ion here. These ions here are very happy in their ionic lattice. We've got positive ions next to negative ions, and they're pretty happy like that. So why do they enter into solution? Well, there's a small positive charge on that part of a water molecule, a small negative charge on this bit, and so they're able to cluster around a negative ion like this, or around a positive ion like this. And what this means is that ions can be just as happy in solution as they can be as a crystalline structure. And really that's all we need to know at iGCSE level as to why things dissolve. Now, why are we talking about that now? It is because we're going to be thinking about how this affects the concentration of water molecules. So let's take this solution here in this whole area. We have quite a lot of these water molecules here. Those would be called free water molecules because they're not attached to any ions like this. And our concentration of free water molecules goes down as we dissolve more things in that water. As more of these water molecules are used up by being bound onto solute molecules, so our concentration of free water molecules goes down. So our principle is, as solutes dissolve, water concentration falls. Okay? As solutes dissolve, water concentration falls. That's our first principle. Let's take that forward. This is a diagram you may well be familiar with. It has here a semi-permeable membrane. And this semi-permeable membrane has a particular property that it allows water molecules to cross, which are small molecules, and it'll allow other things to cross, such as O2 and CO2, but it will not let larger sucrose or Na+ or Cl- or any of these ions, it will not let these larger molecules cross over. They stay on one side. So let's apply the principle of what we learned on the previous slide to this slide here. This here is pure water, and here we have a sucrose solution. The concentration of this right-hand side of water molecules is lower. So the conc of H2O is lower. Why? Well, because we have these solute molecules dissolved in it. Therefore, the concentration on this side is higher of water molecules. Higher water concentration. Hey, that's not a particular surprise to find that the concentration of water in pure water is higher than the concentration of water in a sucrose solution. That's not going to be a surprise to us. But what is going to be a surprise is the effect that that then has. We know about diffusion already, in that molecules tend to diffuse from an area of high concentration to an area of low concentration. Where's our area of high water concentration? Our area of high water concentration is on the left-hand side here. Where are we? Here we go. So high water concentration on the left-hand side here, and so molecules will tend to diffuse in this direction. Yeah, we'll get a couple coming back like that. But on balance, our water movement is from left to right rather than from right to left. This is osmosis. This is the diffusion of water molecules across a semi-permeable membrane. And it will happen from a higher water concentration to a lower water concentration. However, at this point, we're going to change the phrase water concentration to the phrase, I'm going to write it in the top right here, to the phrase water potential. This is to avoid confusion. Water potential is equivalent to water concentration. Why don't we talk about water concentration? Really, because if you're talking about a solution, you say you have a highly concentrated solution, that probably means it's got a low water concentration, and if you have, well, you just get confused as to what concentration you're talking about. So we talk about water potential, and you can think of it as being equivalent to water concentration. As you dissolve things into water, this lowers the water potential. So more solutes, lower water potential. And pure water, here we go, pure water has a water potential of 0. A water potential of 0, and any solution, that is water with things dissolved in it, has a negative water potential. And water will diffuse, as we've discussed, from an area of higher water potential, 0 in the case of the left-hand side of this, to an area of lower water potential. That is the negative water potential on the right-hand side. Well, let's have a look at a definition of that, and then we'll see how it affects cells. Here's our written definition. Osmosis is the water of water molecules from an area of high water concentration to an area of low water concentration across a selectively permeable membrane. Yes, we've called it a semi-permeable membrane. You can also call it a selectively permeable one. And we would now substitute the phrase water concentration for the phrase water potential. Osmosis in cells. The water potential of a cell is crucial to its function. We'll think first of all about animal cells. Cells are big sacs of solutions. So if we take this cell here, we have a solution, that is the cytoplasm, surrounded by and we'll call it selectively, why not? A selectively permeable membrane. And because of this, this cell is going to be affected by osmosis. Let's say we put an animal cell in pure water. What's our water potential outside? Well, it is going to be 0. Therefore, a high water potential. Inside the cell is a solution. The cytoplasm is a jelly-like solution. It contains all sorts of things like glucose and ATP and proteins and lots of wonderful things like that, and amino acids. And all those solutes are going to lower the water potential. So we're going to have a lower WP water potential in here, and therefore water, as this arrow indicates, is going to move in by osmosis. That's all well and good, except the trouble for an animal cell is that it does not have a cell wall, and therefore as it expands and expands, as water enters it, it will burst. And our term for burst is to lyse. It becomes a lysed cell. Let's see what would happen if we put it in a concentrated salt solution. So, in here we still have our low water potential, but if we have outside a very low water potential, the water potential in here, i.e. the concentration of water molecules, will be higher than that outside, and so water will move out by osmosis. There it is, and it will become all shriveled up like that. Another word for this is crenated, not cremated, crenated. This again is going to be very important because the enzymes within the cell will rely on working at a certain concentration, and with the cell having a certain concentration of water inside it. And so this also is going to be bad news for an animal cell. Animal cells love to be in a solution which has the same water potential as their cytoplasm. If they do, then water will neither move in nor out. That will be important for an animal cell, and that's why they have the same water potential as your blood. Let's think about it in plant cells now. Let's take one and surround it with pure water. Again, out on the outside we will have a high water potential, and on the inside we will have, well, let's call it a medium water potential. As a consequence, we've got a higher water potential outside than inside, and therefore this will increase in mass as water goes in. It will also cause our vacuole, this is our vacuole here, to swell up, and you'll be able to see that nicely under a microscope. In fact, that vacuole would probably be much larger in this cell. You'll see that as lots of water enters into this, it kind of pushes against the inside of the wall. So constantly we've got this pressure now pressing out onto the inside of the wall. By the way, let's remember where the cell membrane is. That is the cell membrane on the inside there. That is functioning as our selectively permeable membrane. And as this pushes out, it generates a pressure pushing out, and then the wall itself will push back in to counteract this because it's slightly stretchy, and this is called turgor pressure. You can think of it as being a little bit like a hot water bottle, which is totally full with no air bubbles in it at all, and they're pretty firm, pretty rigid, and that's what a plant cell will be like when you put it in pure water. That cell is said to be turgid. Great. Let's have a look at what happens then if we put it in a salty solution. I know it says sugar, but hey, sugar, salt, a strong sugar solution. Well, in here again we have our medium water potential, and out here we have our low water potential, and therefore water is going to move down a water potential gradient. By the way, what do we mean by a water potential gradient? Well, if I have a high water potential here and a low water potential here, we can join those dots, and that slope is a gradient. And so when we talk about moving from a high to a low water potential, we're talking about moving down a water potential gradient. So water then moves out of this cell across its selectively permeable membrane. Why have I drawn the arrow only going that far and not going past the cell wall? Well, for a start, I could have drawn it going past the cell wall, because it will, but also because osmosis is happening across this selectively permeable barrier here. This is the selectively permeable barrier, and as a consequence, it will allow osmosis to occur. The cell wall is not selectively permeable. It's pretty much permeable to everything, so that doesn't really affect osmosis. Now what that means is that the solution out here diffuses in here, and these gaps here, this is solution. This is not air or anything like that. That is the solution. So what is happening is that the water is leaving the cell by osmosis, and then the cell wall can't really bulge in to meet the now shriveled cell on the inside, and so the cell membrane pulls away from the cell wall, and then behind it comes all this solution to fill the gap. So the cell membrane, it just holds on to certain pinch points here and here. For the sake of the word, those are called plasmodesmata, and they just hold on there. That is a plasmolyzed cell, and it's going to be floppy, and it's not going to be very much use in structural support for a plant. And so if a plant loses a whole lot of water, its water potential is going to fall inside its cells, it's going to lose a lot of turgor pressure, and it's going to become limp as well. And these cells are said to be plasmolyzed. Poorly underlined there. Now, turgid cells support plants, and can I recommend having a little look at this YouTube clip? I'll put a link underneath this video to this, and you can watch a basil plant wilting. I know that you want to do that. So that's it. That is how osmosis affects animal and plant cells. Here are some tutorial questions to finish off this video. Question number 1. What is water potential? Can you explain that and define it? Question number 2. Why do solutions have a negative water potential? Question 3. Can you explain why a carrot will gain mass when left in a bowl of water? Number 4. Explain why a basil plant will wilt if you water it with a salty solution. And question 5. Can you explain why it is important that the blood plasma has the same water potential as the cytoplasm of our cells? Indeed, the word to use there is isotonic, and that means having the same water potential. Thank you, and I hope that's helpful.